The overall goal of this procedure is to generate Epstein-Barr virus-transformed lymphoblastoid cell lines, or LCL, from human B cells. This is accomplished by first isolating peripheral blood mononuclear cells from human blood. Next, the cells are infected with Epstein-Barr virus in the presence of FK506, an immunosuppressive agent. 3 to 4 days after infection with Epstein-Barr virus, a subpopulation of cells emerges that is marked as CD23 high, CD58 positive, and is predicted to undergo proliferation. CD23 high, CD58 positive cells can be enumerated by flow cytometry as a predictor of successful outcome. Proliferating cells can be further identified micro and macroscopically by the clusters of lymphoblastoid cell lines that form in successfully transformed cell cultures. The final step is to expand lymphoblastoid cell lines for cryopreservation.
In my laboratory, we generate lymphoblastoid cell lines, or LCLs, to understand interactions between the host and Epstein-Barr virus. Lymphoblastoid cell lines are also of broad general interest to the scientific community, particularly to virologists, immunologists, and cancer biologists.
As a postdoctoral fellow in the Baduri McIntosh lab, I'll be demonstrating the procedure for generating lymphoblastoid cell lines.
After drawing 10 ml of blood from a donor, add the heparinized blood to a 50 ml conical tube, and then dilute it 1 to 2 by adding 20 ml of room temperature PBS to the tube. Now underlay the diluted blood with 15 ml of Vicol hypaque lymphocyte separation medium, and then spin down the blood at 225 times G without the break at room temperature for 30 minutes to establish a gradient. Next, remove the buffy coat and transfer it to a new 50 ml conical tube. Bring the volume up to 50 ml with PBS and then pellet the cells at 600 times G for 10 minutes. Pour off the supernatant, resuspend the pellet in 50 ml of PBS and then wash the cells 2 more times under the same conditions. Now resuspend the cells in 1 ml of complete RPMI. Use 5 microliters of cells to prepare a 1 to 10 dilution in trypan blue and count the live cells using a hemocytometer. Then use complete RPMI to adjust the volume to obtain a cell concentration of 2 times 10 to the 6 cells per milliliter, and transfer the cells to a 25 cm squared tissue culture flask. To prepare the cells for infection with Epstein-Barr virus, add FK506 to the tissue culture flask to a final concentration of 20 nanomoles. Then, place the flask in a 5% carbon dioxide incubator at 37 degrees Celsius for 1 hour. When the cells are almost finished incubating, rapidly thaw an aliquot of Epstein-Barr virus harvested from B95 8 cells. Then, remove the flask from the incubator and add the Epstein-Barr virus to the cells at 1 to 10 dilution. Now swirl the flask to mix the Epstein-Barr virus into solution, and then place the flask upright in the incubator with the cap on but slightly loose if using a non-vented flask for 2 to 3 weeks prior to expansion of the culture for cryopreservation. A week after Epstein-Barr virus infection, clusters of cells are visible by light microscopy, as seen in this representative figure of early microscopic clusters. As time progresses, the microscopic clusters become larger, such that clumps are visible macroscopically in the flask, as seen in this figure, where larger clusters of LCL can be observed. To feed the cells, double the volume of the supportive medium in the culture flask on day 12. When the culture medium turns yellow, typically about once a week, it is generally time to feed the cells. To expand the cells, increase the volume of the culture 2 to 3-fold using complete RPMI. Expand the culture to 70 to 100 milliliters in a 75 cm square flask over the next few weeks to generate cells for cryopreservation. A successful outcome is predicted by the presence of CD23 high, CD58 positive cells. As seen in this scatter plot, approximately 2 to 3% of live cells on day 3 to 4 after exposure to Epstein-Barr virus have this profile. Other subpopulations of CD23 positive, CD23 negative, CD58 positive and CD58 negative cells observed after exposure to Epstein-Barr virus show minimal proliferation. And uninfected cells, or cells infected with a non-immortalizing strain of Epstein-Barr virus, do not demonstrate CD23 high, CD58 positive cells. The cells may also be visualized by light microscopy. As mentioned, within a week of Epstein-Barr virus infection, small clusters of cells are visible in the flask by light microscopy. As time progresses and the cells develop into LCL, the microscopic clusters become larger, such that clumps are visible macroscopically in the flask.
After watching this video, you should have a good understanding of how to isolate peripheral blood mononuclear cells from human blood and to generate Epstein-Barr virus transformed lymphoblastoid cell lines from human B cells.
While generation of lymphoblastoid cell lines has been described by other laboratories, the advantage of our protocol is that it provides the researchers with a method to predict successful outcome as early as 3 to 4 days after infection of B cells with Epstein-Barr virus.